afternoon. I'm happy to introduce Ryo Ishibashi from Cho University. He's presenting tensor decomposition based unsupervised feature extraction with optimized standard deviation applied to differentially expressed genes, DNA methylation, and histone modification. Thank you.
Hello, thank you for coming to my presentation. I'm Ryo Ishibashi, and a second-year master's student. I belong to the Department of Physics at Chu University in Tokyo. Today, I'd like to introduce a research of my supervisor, Professor Taguchi, on his behalf. This is the title. This presentation has already been preprinted. You can access it here, and this slide is available via this QR code. First, I will touch on our claims. First, our identification of DEGs, differentially expressed genes, using PCA and tensor decomposition leads to better result than existing method by optimizing standard deviation used to generate the null hypothesis. Second, as well as DEG's identification, our method can be applied to histone modification and maturations. This is today's outlines. For detailed explanation, please refer to the preprints or this Springer book. First, background, large P and small n problem. In general, genomic data is huge, while samples such as patient and cell are few, so it is difficult to analyze. Also, individual methods have been proposed for certain experimental data, such as histone modification and DNA methylation. So today we propose a tensor decomposition method that can be applied to various types of experimental data. Next, why do we choose PCA and tensor decomposition? These are the reasons we choose PCA and tensor decomposition. The first is ability to handle a variety of data. Second, interpretation is easy. Third, it is easy to program and computationally inexpensive because both PCA and tensor decomposition are unsupervised learning. Next, what is a tensor decomposition? A higher order singular value decomposition over tensor is specific orthogonal Tucker decomposition. And as the name implies, it is an extension of singular value decomposition to multiple dimensions. This means that important features of the original data can be extracted. And in this study, higher order singular value decomposition enables to find DEGs by selecting the desired features. This is how to interpret tensor decomposition. For example, if we consider the third order tensor of gene and sample and tissue, UL1I is gene-dependent vectors, UL2J is sample-dependent vector, UL3K is tissue-dependent vector. We'd like to find DEGs, differentially expressed genes, that differ between a patient and a healthy control and tissue-specific. So we first find sample-dependent vector such that there is difference between patient and healthy control. Then we find the UL3K tissue-dependent vector, which has differences among the tissue. Finally, we can identify the DEGs by finding L1 that maximize core tensor max Z from identified L2 and L3. This is because the larger the value of core tensor, the larger the contribution to the original tensor. Finally, analysis procedure. After principal component analysis and tensor decomposition, we can select the sample-dependent vector and tissue-dependent vector that has the desired properties. Then the gene vector is determined. Here for the gene selection criteria, we assume that a gene-dependent vector U follows Gaussian distribution and we assign a p-value, the independent vector u in the cumulative chi-square distribution. When following a Gaussian distribution perfectly, this 1 minus p histogram is flat, except around 1 minus p equals 1. However, in many cases, it is not and is disordered. So in this study, standard deviation is optimized by excluding outliers to follow Gaussian distribution. I will explain about it later. Finally, result, benchmark dataset. MAQC is frequently used for a benchmark study. We analyze RNA-seq. Xij represents expression of ith gene at jth sample. Samples are 7 universal human reference RNA and 7 human brain reference RNA. The MAQC data looks like this. The density distribution shows that some genes are common, like this, and some are different among the sample. So we apply principal component analysis to the MAQC data and look at sample-dependent vector V1 and V2. V1 is the mean vector with no difference among the sample. On the other hand, V2 is a vector with a difference among the sample, like this. Next, this is scatter plot of the gene-dependent vector U1 and U2. We can see that mean genes are clustered in one place at this point. Then we assign a p-value to gene-dependent vector U2. On the left is 1 minus p histogram. On the right is 1 minus p histogram with standard deviation optimized to follow Gaussian distribution as much as possible. After optimization, the histogram is closer to Gaussian distribution. This means that outliers can be successfully considered outliers. Finally, a gene with p-value less than 0.1 were selected. Furthermore, in general, genes with high expression are more likely to be DEGs, differentially expressed genes. Existing methods take this empirical rule into account. However, we do not need it and can select reasonable genes. The selected genes were uploaded to Enricher for enrichment analysis. The gene selected by our method are better than existing method. In conclusion, our method outperforms various state of the art method while assuming neither empirical dispersion relation nor negative binomial distribution. Next, DNA methylation and histone modification. I don't have enough time to explain about DNA methylation and histone modification. So if you are interested, you can access more detailed here. So in conclusion, our method also outperformed existing method for DNA methylation and histone modification. Finally, conclusion. These are the main conclusions of the study. First, principal component analysis based on the tensor decomposition based on supervised feature extraction applied to the identification of differentially expressed genes, DEGs, can outperform various states of the art method, including the SEC2, when standard deviation used to generate the null hypothesis are optimized. Second, they are applicable to the identification of differentially methylated cytosine as well as differential histone modification without specific modification as well. That completes my presentation. Thank you.
Okay, given time constraints, we're going to not take any questions, and we'll be ready for our next speaker, Tiana Milinkovic.